Alright, good day everyone. Today we're going to start with chapter 1. Chapter 1, it is about expression of biological information. Okay, so let's start with the first slide. What carries the genetic information? In the past, people used to think that protein is the one that carries the genetic material. Because you look at the father organism and the offspring and the grand offspring, they look similar. So meaning that what makes the youngest one look just like the previous generation? So there must be something being transferred. So why do they think proteins are the one carrying this information? There's a lot of type of protein. They're more structurally diverse. And the thing is, DNA content, if you want to compare with protein, they are found to be the same in the different cells of organism, but RNA and protein are different. The thing is, if you look at the cells of an organism, they have a lot of cells. You have the lung cell, you have the skin cell, you have the eye cell, you have the brain cell. Okay, all of them have different number of proteins and RNAs, but the DNA content are all the same. Okay, so it is also found out that somatic cells of the plant organism contain twice the amount of DNA compared to germ cells. And more RNA proteins were found in cytoplasm, which means the possibility of DNA being genetic material was higher. So there are some early and puzzling clues. In 1800s, Marshall found DNA in nuclei. Okay, so he found this chemical called DNA. Early 1900s, Griffith transferred hereditary material from dead cells to live cells. There are two types of bacteria that kills in mice. So you have the bacteria with the R cells. R means rough. This bacteria will have a rough outer membrane. It means that it doesn't have any protection against the antibody of the rat. So when you inject into a rat, the rat lives because the rat's antibodies would be able to kill that bacteria. If you want to compare it with the S is a smooth bacteria, which means that this bacteria would have the shield around it that makes it look smooth. This shield will be protecting them from the antibodies. So when you inject it to mice antibody cannot kill it, but the bacteria, because it's protected, it can kill the mice. So what did Griffith do? That he mixed both the S cells, which are dead, with the live R cells. So somehow, in this mixture, if this mixture is put into the mice actually died, just like how it did with the S cells. So after that, they did a post-mortem, in that you open up the dead mice. They found out that all of the bacteria that used to be R now it's become S. So from the rough, unarmored bacteria, it becomes a shielded, smooth bacteria. So they transformed. So the dead cells, somehow they transformed the live cells. So Avery and McCarty find the transforming principle. In 1940, Avery and McCarty separated the S cells from Griffith's experiment into lipid, protein, and nucleic acids components. When lipid, proteins, and RNA were destroyed, so the remaining substance, DNA, still transformed R cells to S cells. So it means that what is the thing that transformed them from a harmless bacteria which are unable to fight against antibody to a shielded, strong, dangerous bacteria that's able to attack and kill the rat cells? It will be the DNA. So conformation. Hershey and Chase in 1950, they experimented with bacteriophages. Bacteriophages are the viruses that can infect bacteria. So if you can see in this experiment, they labeled sulfur with something like a radionuclide. So I mean that, sulfur is 32, right? If you put a radioactive sulfur, it is a little bit heavier. Okay, 35S, means that you can detect with the radioactive. And then the DNA has phosphorus, so they tag this DNA with a heavier phosphorus, which is 32P. Okay, so they took the viruses which has both been marked and let them infect this bacteria. Okay. So what happens after that is that they found out that within the cells there is no sulfur inside, but there is sulfur outside of the cell. Okay. And then they also found out that within that same cell there's a lot of phosphorus. So meaning that the phosphorus from DNA core actually get inside of the bacterial cells and remain there. So meaning that the thing that changes the bacteria will be the DNA, not protein. Okay. So discovery of the DNA structure, this one actually not just these two guys, but actually has done by many different scientists. Okay, more than a century of work has been put into this to find the shape of the DNA. So Watson and Crick discovery of DNA structure was based on almost 50 years of research of other scientists. So here is Rosalind Franklin. Rosalind Franklin was the first person who found this X shape. Okay, so what is the X shape? This X shape is what you call the shadow of the DNA. So the thing is, DNA is a molecule that we cannot see with our own eyes. So what she did was she did X-ray crystallography. She shined X-ray on the molecule, and then what you can see is the shadow of the molecule being reflected on a crystal. So they found out that it is a helix. So this knowledge was used and added to an existing research done by Watson and Crick. Based on this, it is the final piece of evidence that they will build the model of the DNA. Okay, now for DNA building blocks, DNA building block is made out of nucleoside. So DNA is actually a long chain. Each one of the links of the chain is a nucleotide. And what are nucleotides? You can see this is a nucleotide. It has nitrogenous base, sugar, and phosphate group. So phosphate group and sugar will always be same, but the nitrogenous base can change. It can be either adenine, thymine, cytosine, or guanine. Okay, so here is Chargaff's first rule. Okay, it means that A must be same as T, G must be same as C. So the amount of thymine and adenine in DNA must be same. The amount of purines and pyrimidines, G and C, must also be same. Chargaff's second rule is called the second parity rule. So in 1968, Chargaff discovered that in Semiconservative DNA, A would pair with T, C would pair with G. Okay, so according to Watson and Crick's DNA model, the DNA molecule looks like a staircase, but it actually has two axes. So it is a double helix. A single helix is just like a staircase, in that you have something going around a single axis. But here you can see there is two axes, the large axis and the small axis, right? So this is what you call a double helix. So they will coil around each other, and then the bases will pair, A with T, G with C. So the two strands of the DNA are anti-parallel. What does it mean? It means that it goes from the 3 to 5 in one end, and in the other strand from 5 to 3. What are the numbers 5 to 3? It is actually the carbon number on the sugar. Remember that the sugar looks like this pentagon, right? 1, 2, 3, 4, 5. Okay, 5 is up. Okay, in another configuration, in another strand, the third sugar is up. So each strand will be very long, up to millions of nucleotides, and these two strands are linked with hydrogen bonds. Hydrogen bonds is very stable. Okay, so now we move to DNA replication and repair. DNA would be copied before mitosis or meiosis 1, and DNA repair mechanisms and proofreading correct most replication errors. Okay, so the thing is, there is this term called semi-conservative. So the thing is, you have two strands of DNA. So the new strand will be made based on one strand of the two. So meaning that when new DNA are made, one is the old, one is the new, because the old DNA is used as a template to build the new DNA. So that's what you call the semi-conservative. If it is totally conservative, means that the new DNA will just be new molecules all over and no old portions. But in here, in the semi-conservative DNA replication, the new one will be made based on the old one. So the new DNA will have one old, one new. Okay, so in this picture, you can see that there's forming a replication bubble, imagine that you have a zipper, right? The zipper will open in two places, here and here, but the building of DNA can only go from the 5' to the 3'. Okay, so what happens that you have little by little you have a gap here. Okay, so these are called the Okazaki fragments. Okazaki fragment will soon combine into a long strand. So as you can see in this picture right here, this is the parent, right? The parent DNA, the original, the old DNA in blue. And the old DNA will unzip, so the new DNA will be made based on the template in the old DNA or the parent DNA. Now you have the new DNA made out of two. One is the parent strand, one is the daughter strand. So here are the DNA replication enzymes. You have helicase. Helicase means that it is anti-helix. It doesn't want to helix, so it will open the bonds between DNA strands, so the strands are not helix anymore. And then you have the DNA polymerase. Polymerase means that it makes new DNA molecules. And then you have the DNA ligase. Ligase will join or glue together the DNA segment or discontinuous strand. Okay, primase. Primase will be creating a short segmented RNA primer for attachment of DNA polymerase. Meaning that DNA polymerase, it is an enzyme that needs a place to start. So the primase will be like a marker saying here is the entry, enter here. So polymerase will go there and start the building of new DNA from there. The nuclease. Nuclease will cut the phosphodiester bonds between nucleic acids, checking for mistakes. So you have DNA repair mechanisms. When DNA polymerase proofread DNA sequences during replication, okay, it will do something that's called repair. So let's say that your DNA gets bombarded by a lot of X-ray, a lot of gamma ray. It will damage your DNA. Suddenly your nucleotide will just be destroyed. So you have empty space right here. Okay, so the proofreading and repair mechanisms will detect, oh, there is a empty space, and so they will repair the DNA using DNA polymerase. And then this nick or this small gap is closed with DNA ligase. But the thing is, sometimes they can fail, and when they fail, you will have a mutation. The error becomes mutation. Mutation is a permanent change in the DNA sequence. Application of DNA replication. So you have cloning. So cloning means you want to make a new organism that is exactly the same as the adult individual, which is the original. Okay, so it is an exact copy of a molecule, cell, or individual. It occurs in nature by asexual reproduction or embryo splitting. But there is a technique that is used by humans called reproductive cloning methods. Okay, the first technique is called somatic cell nuclear transfer. So you know that for ovum, right? Ovum is the egg cell. Within that ovum, you have a nucleus which is just half of the chromosomes that the human has. So we take out that incomplete nucleus, we put in a complete nucleus which is from a somatic cell. You know, other cells in the human body, they have complete chromosomes. We put it in that egg cell. So now it can actually become a new organism. Or we can use something that's called induced pluripotent stem cell, iPSCs. So pluripotent means it can become anything. Plural, potent, potential, right? Okay, so here is SNT. In SNT, nuclear DNA of an adult is transferred to an enucleated egg. So we take out the nucleus from an egg, ovum. We put the nucleus of the somatic cell into that nucleus, into that egg cell. Egg cell represents reprograms differentiated DNA to act like undifferentiated DNA. So what do you mean? It actually becomes a hybrid cell. The hybrid cell will develop into an embryo that is genetically identical to the donor individual. So meaning that the clone will have the exact same nucleus just like the original. So therapeutic cloning, okay, if you look at the name, therapeutic means something that you use to cure diseases, right? Therapeutic cloning means that you use it to cure diseases. Okay, you create human embryos for research purposes. It doesn't mean that we create a baby, right? We just want to create a certain cell to replace the cell that we lost or destroyed because of a disease. Because now it is like a new cell, right? We can use that to make a skin cell, to make a nerve cell, to make a red blood cell. Okay, we call it cells for therapy. So technically, they are embryos. It's just that they are in a petri dish. Okay, they are not babies, but they are just a bundle of cells that we harvest. Okay, right. Here is from DNA to protein. Okay, so here is the nature of genetic information. What is genetic information and how does the cell use it? As you can see here, this whole thing right here is the DNA strand. Okay, this DNA strand is something like what you call a recipe, right? Recipe book. You read it, and then you know what to build from the recipe. So, compared to us, English has how many letters? 26 letters, right? A to Z. But for the DNA, the language only has 4 letters, and that 4 letters is enough to create all of the protein that we need for life. Okay. So first of all, this DNA must be transcribed into RNA. Transcribe means you are copying exactly the same thing from a source. Okay. So let's say that you have a recipe book, right? You want to make a cake. Okay. You get a book and then open the chapter called How to Bake Cake. So what you do is that because you are in the library, you cannot take it out of the library. Okay. What you do is you take a paper and then you write down the recipe. So that's how transcription is. So transcription means that you make an mRNA that is based on the DNA of interest. So let's say that the cell wants to make some enzyme. So the gene that codes for the enzyme will be transcribed into the mRNA. Okay. So if we look at here, the DNA is open, right? It will show all of the bases A, T, G, C, T, A, G, G, C. So what happens is that mRNA will be made based on this sequence. It's just that for mRNA doesn't have a T, it has a U. So instead of like A, T, you have A, U. But others are the same. So messenger RNA would have the information transcribed from DNA. Ribosomal RNA is different. It is the main component of ribosomes where polypeptide chains are built. So ribosomes are, remember, they are the factory within your cell. They create proteins. And transfer RNA or tRNA, they deliver amino acids to ribosomes, meaning that they will bring the amino acids into ribosomes so that the amino acids can be built into proteins. So converting mRNA to protein. Okay, so as you can see here, this is what you call the central dogma. DNA to RNA to protein. Okay, so from RNA to protein is a little bit different. We call it translation. Okay, so you have that recipe. It's now time for you to make that cake. Okay, so gene expression, if you can see here, is how the information within the DNA is turned into a product. You read a recipe, you make something from that. So it actually takes multi-step process: transcription, translation. Okay, and then that translation will build something, a protein. The protein will build a cell. A cell will build a tissue. A tissue will be an organ. An organ becomes a system, right? So it is a multi-step process. It means that if you change somebody's DNA, the person doesn't change, instantly. It actually takes time for them to change. So here, transition DNA to RNA. First of all, RNA polymerase assemble RNA by linking RNA nucleotide to chain. Okay, based on the DNA sequence. So the RNA sequence will be just exactly the opposing nucleotide compared to the DNA. So what about replication and transcription? In replication means that you make a new DNA that is exactly same as the old DNA. You know, replica. The thing is, replication and transcription have the same process. You zip open the DNA, and then the RNA polymerase makes a strand based on that old DNA's open sequence. Okay, as you can see here, it opens, and then they will make a new strand based on the nucleotides A, T, C, G, A, G, A, right? So how does it start? It starts with, first of all, you must have a promoter. Promoter is the start point. Okay, so RNA polymerase will bind to promoter, and promoter will be the start point. Okay, and then RNA polymerase will start to unwind the DNA. The DNA will no longer be like this. It will be unzipped. When it's unzipped, then RNA polymerase can make an RNA transcript. So RNA polymerase will move all the way to the end, and as it moves, it will add more and more and more longer RNA. Do you remember when you go to the shops, right? The cashier is printing the receipt. It's kind of like that, and then the receipt goes up all the way out of the cashier machine, something like that. Okay, so a long RNA will be created, right? So the RNA polymerase will do that until it reaches this terminator or termination point. At this point, RNA polymerase will say, okay, this is the end, I will stop here. So when it stops there, you have the completed RNA transcript. So after transcription, RNA will be modified before it leaves nucleus in a mature mRNA. So there might be some useless parts of the RNA that is not important to make protein. Okay, for example, you have something that's called intron and exon. Introns are the gaps that do not code for any protein. Okay? Something like if you read a book, right? They are kind of like empty pages or a title page, okay? Or maybe something like you know, something like in between page, right? They just shows you chapter 1. Okay, it doesn't have anything inside for you to read. Okay, so those are the parts that we do not want, so those are removed. So the only parts that is left will be the exons. Okay, so the thing is, cutting exon and intron doesn't actually happen the same way, you know, because you know when you read the book, sometimes you just skip. Okay, and in other times you don't skip it, you just read, and then you'll be like, okay, I know that's what I want. So this is what you call alternative splicing. So let's say we have this gene 1, 2, 3, 4. Okay, 4 exons. You take away the introns, so you have 1, 2, 3, 4 in the mRNA. So what happens that after that the mRNA can be modified to have just 1, 2, 3 or 1, 2, 4. So this 1, 2, 3, maybe like you read a book, right? You read chapter 1, chapter 2, and then chapter 3, and then you say, okay, I know the ending. Okay, I can actually do something with this knowledge. Okay, I can bake that cake. Okay, but if you read chapter 1 to 4, you skip 3, and then you go, okay, I can make a cake from this, and then you make another different cake, right? So that's how it happens. Because of the alternative splicing, it can create different proteins. After splicing, transcripts are finished with the modified guanine cap at the 5' end and poly A tail at the 3' end. At the 5' end, you have this guanine cap, 3' end poly A tail. So splicing gives the sequence, and modifications increase stability, so it will not break. mRNA, if you look at the name messenger RNA, it is for carrying information from the nucleus all the way to the ribosomes. You know that for the letters, alphabets in the RNA or DNA is just 4, A, T, C, G. Okay, what about the words? The words are just 3-letter words. You know how many 3-letter words that you can make? DOG, dog, CAT, cat, yeah, APE, ape, right? Okay, it's kind of like that. So within the mRNA, you have a lot of 3-letter words made out of these 4 alphabets. So you have ATC, TAG, TAT, TTT, something like that. Okay, so that's what we call a codon. A codon is a sequence of 3 nucleotides. Okay, so as you can see here, the base triplets in all mRNA are words in the protein building message. So AGC, you make this. UGA, make this. ACC, make this. Okay. What happens that is within the ribosome, this thing right here is called the transfer RNA, tRNA. tRNA will always be attached with an amino acid. So based on the message on the mRNA, the tRNA will come and match itself to the codon in the mRNA and attach the amino acid that it carries to the existing chain of amino acid. Genetic code. It consists of 64 mRNA codons, meaning that this is the whole dictionary of the codons. Okay, some amino acids can be coded by more than one codon. Some codons signal the start of end of the gene. AUG is for the start codon. UAA, UAG, UGA are stop codons. So here is the genetic code. Okay, as you can see, UUU creates this. UUA creates this. Okay, UCUU creates Liu. Okay, just like CUC, CUA, and CUG. Okay, if it goes to UAA, UAG, or UGA, it will cause the RNA polymerase or DNA. It will cause the ribosome to stop building the amino acid. Okay. So here is the tRNA, the translators. The tRNA delivers amino acids to ribosomes. It has an anticodon complementary to an mRNA. So if a mRNA is AUU, it means the tRNA must be TAA. It is anti of the codon, and also it carries an amino acid attached to it. So each one of these amino acid is carried based on the anticodon that it has. Okay, each amino acid will be carried based on the codon that matches with the anticodon. CCC, alanine. Translation will convert the genetic information that is in mRNA into a new protein chain. So the thing is, a lot of things that is in your body are made out of proteins. So this here is how the building blocks are made based on the information that is in mRNA. Translation occurs in 3 stages: initiation, elongation, and termination. You have initiation. Initiation means the starting. So small ribosomal unit binds to mRNA like this. Anticodon of the initial tRNA was base pair with the start codon. So start codon will always be AUG. AUG is start codon. A large ribosomal subunit joins the small ribosomal subunit. So the thing is, small and big ribosomal subunit will be like a snowman attached to the mRNA. Elongation. mRNA moves to the ribosome via the A, P, and E sites from 5 to 3. As you can see, this is the snowman shape. So you have A, P, E sites. Okay, so first of all, mRNA starts with A. Okay, and if it is in A, tRNA will bind. And then at P, the amino acid will attach or join. And then when it moves to E, it will eject. So like this, and then this. What happen is that this tRNA that has been merged is amino acid with the previous will be removed. Okay. So the chain of polypeptide will grow longer. So when the ribosome encounters the stop codon, polypeptide synthesis ends. So the release factor will bind to the ribosome, and enzymes will detach the mRNA and polypeptide chain from the ribosome. As you can see here, it will detach. So what we have is a long polypeptide here, and here is the unused mRNA. Okay. So now we move to gene regulation. Gene regulation is the control of genes, a process to turn the genes on or off. Okay. So operon is the functional unit of the key nucleotide sequences of DNA. So mean that in operon means that you have a DNA. Okay, this DNA gene do something, and then you have a switch that turns it on and off. So operon must all have that, because you don't want your DNA being turned on all the time. Okay, you don't want to make cake all day long. Okay, eat dinner cake, breakfast cake. Okay, what else? Okay, even going to school, you bring cake. Okay, you don't want to do that. So you just want it for special occasions. So that's how operon works. Operon will only turn on the gene when it's needed. So it has a functional, operator, promoter, and some genes that can be translated into something. Usually genes are turned off when there's no need, but when there is something that needs to be done, it means that they are needed. Okay, for example, you have E. coli. E. coli is this bacteria that doesn't create lactase without any lactose. So how do they find this? Francois Jacob and Jacques Monod demonstrated that genes in E. coli are regulated in the biochemical level. So they have one of the three proteins the bacteria use to obtain energy from sugar lactose. So here is the whole DNA. No, not whole DNA. So here is the operon in the DNA of the E. coli. So you have the promoter, the regulatory gene. This one is a shut off switch, right? Operator is promoter for such a gene. It is the start line for the promoter polymerase. And then you have operator, or they have the three genes that actually do something or create that enzyme. Z, Y, A. Okay. Repressor is the switch. Okay, it turns operon off. Why? Because the repressor will create a protein. The protein will fit on the operator. It will stop any RNA polymerase from coming. Okay, so mean that it will guard the gene from being transcripted. Right? Imagine like you have a recipe, but the recipe book has been locked. There's a padlock. Okay, you cannot access that book at all. Promoter. It is where RNA polymerase attach here to begin transcription of the genes. Okay. Operator is a segment of operon where repressor fits to block RNA polymerase and turn off transcription. Repressor gene creates the repressor protein. The repressor protein shuts off that gene so that you cannot use it. So meaning that the repressor is the padlock, right? A repressor gene is the instructions to make that padlock so that you can access that book. Structural genes are the recipes to make the cake that you want, like Z, like Y, like A. In this case, it is for lactose. So without lactose, the repressor gene will create this red thing called the repressor protein. Repressor protein will clamp onto the promoter side. So RNA polymerase, if it comes and then you'll be like, oh, it's locked, I cannot access. So it's not transcribed. So the RNA polymerase cannot do anything. But with lactose, here you can see the lactose protein, it comes and it clamps against the repressor. So now the switch is not off, it is on. Okay, so since there is nothing binding to the promoter, the RNA polymerase will just, okay, I can make the lactose right now. Zoop, you go and make mRNA to make the lactase. So now the bacteria can get sugar from lactose. So mean that now the promoter has been freed because the repressor protein has been removed. Gene is turned on. The end, and I hope you stay tuned for the next class.